Neuropharmacology is a very important field of neuroscience. It forms the basis of the pharmaceutical industry and our hopes for treating brain diseases. There are many compounds that act specifically at different receptors in the brain. So for AMPA receptors, we have CNQX and NBQX. For NMDA receptors, we have APV and MK801. And for the GABA-A receptor, we have antagonists like picrotoxin, bicuculin, and gabazine. These antagonists have highly specific actions and are extremely useful in the experimental setting, but therapeutically, they've not proven to be useful. In this video, we're going to look at benzodiazepines that have turned out to have interesting therapeutic potential and have been extensively used. Benzodiazepines were first developed in the 60s and were marketed as Valium, also known as diazepam, and since then, many benzodiazepines have been developed with higher specificities and better therapeutic outcome. It's interesting to look at the benzodiazepines because they have an extremely specific mechanism of action and a lot is known about how they function at the molecular level, and that's what we'll explore in this video. Let's begin by taking a closer look at the GABA-A receptor. The GABA-A receptor is a pentameric structure made out of 5 different subunits, each of which is an individual protein that come together to form the ligand-gated GABA-A receptor with the chloride channel down the middle. Each subunit is encoded by a single protein, and there are a number of different genes that code for those subunits. They can be divided into different families. So there's an alpha, beta, gamma, rho, delta, pi, epsilon, and theta types of GABA-A receptor genes, and in general, for most GABA-A receptors, they're composed of 2 alpha subunits, 2 beta subunits, and a gamma subunit. And the gamma subunit can also be one of these rho or other ones in gray. The GABA binding site turns out to be somewhere between the alpha and the beta subunit. And so, in fact, there are 2 GABA binding sites between each of the alpha and beta subunits. The benzodiazepine binding site, however, is at a different location and sits between the alpha and the gamma subunits. And in fact, you need to have a gamma-2 or a gamma-free subunit present in the GABA receptor in order for the benzodiazepines to bind. In terms of the amino acid structure of the GABA-A receptor, they form a large N-terminal region where the GABA binding site is encoded, and also there's a benzodiazepine binding site. We have 4 transmembrane alpha helices, M1, 2, 3, and 4, and this one here turns out to be the pore-forming region here, which then hosts the chloride ion channel. And the GABA and the benzodiazepine binding sites are sitting here on the N-terminal, which is on the outside of the cell. The benzodiazepines don't in themselves activate the GABA-A receptor, so they bind to the GABA-A receptor, but it's at a different site where GABA activates the ion channel. And what the benzodiazepines do is that they increase the affinity of the GABA-A receptor for GABA, and so GABA binds more tightly to it in the presence of benzodiazepines. If we think about the electrophysiology functionally relevant in terms of the iPSCs mediated by synaptic transmission, so that's the inhibitory postsynaptic currents, then we'll see that under control conditions, the iPSC has a certain amplitude and a certain duration. And if you then apply benzodiazepine, that then doesn't change the amplitude of the iPSC, but it prolongs the duration of the iPSC. And so the total charge transfer that's mediated in the presence of benzodiazepines is larger, and so it potentiates the GABA-A currents and potentiates the inhibition in the brain. And so benzodiazepines have an interesting action. They don't in themselves cause inhibition, but they only potentiate the effect of GABA when it's normally released in the brain. And so they only act when the GABA is being released at the right time and the right place in the brain. And so it's a highly specific potentiation of GABAergic inhibition that the benzodiazepines mediate. There's further specificity as to which type of GABA A receptors the benzodiazepines act upon. And it turns out that GABA A receptors need to contain alpha 1, 2, 3, or alpha 5 subunits. And if instead they have an alpha 4, alpha 6 subunits, then the benzodiazepines don't have any effect. Now, as the molecular biologists were beginning to clone and sequence the different GABA receptor alpha subunits, they found that there was in general very high homology, so that the amino acid sequences of the different alpha subunits were very similar, but there were of course also many differences. And here in one particular sequence alignment of the different subunits, we see that the 4 that bind benzodiazepines have a histidine at this position 101 for the alpha 1 and alpha 2 subunits. So these are the numbers from the N-terminal, counting from the N-terminal, the amino acids. So we have the different amino acids here. And at position 101 in both alpha 1 and alpha 2, we have a histidine. Alpha 4 and alpha 6 in this equivalent position have an arginine that's present. And so there's a difference in the amino acid structure of the receptors that bind benzodiazepines and the ones that don't. There are, of course, other differences in the amino acid sequences of the GABA A receptors, but we can at least test to see whether this makes the difference for benzodiazepine binding. So we can take the alpha 1 or the alpha 2 subunits and change this histidine to an arginine and now see, does that make a difference to the benzodiazepine effect? Now, the molecular biologists have figured out how to make changes to the amino acid sequence of GABA A receptors, and they've also figured out how to express them in heterologous systems. And so here we're expressing wild-type GABA alpha 1 subunit together with a beta and a gamma subunit, express them, apply GABA, and we see a nice GABA-mediated chloride current. In the presence of diazepam, that's the benzodiazepine Valium, so we apply that at 1 micromolar, and now, when GABA is applied, the GABA-mediated currents are much larger. That's the potentiating effect of benzodiazepines upon the GABA A receptor. Now, in the mutants, where we take the histidine at position 101 and change it to an arginine, that's what's typically present in the alpha 4 and alpha 6 subunits, these are the ones that are insensitive to diazepam. Now we change just this one amino acid in the alpha 1 subunit, and now, when you apply diazepam, there's no potentiation of the GABAergic currents. It's the same in the presence of diazepam as it is without diazepam when this point mutation has been carried out. And so it turns out that this histidine in position 101 is essential in order for the diazepam in order to mediate its effect at potentiating the GABA A receptor. Interestingly, this point mutation makes no difference to the currents evoked by GABA in the absence of diazepam. And so, in many respects, this point-mutated GABA alpha 1 receptor is identical to the wild type, except in the presence of the exogenous agent diazepam or other benzodiazepines. This then allows us to do very interesting genetic testing to see which subunits mediate which actions of the benzodiazepines. So in the mouse genome, we can also go and make exactly the same mutations to the GABA A receptors. We can change in the alpha 1 or the alpha 2 subunits. We can take that histidine at position 101 and change it to an arginine, and then we'll make GABA receptors for these specific subunits that are insensitive to diazepam. And what's good about doing this at the genome level in mice is that we can then see what are the behavioral effects of applying the drug, the benzodiazepine, to an animal in terms of the behavioral change that diazepam makes. And we're interested in two different effects of the benzodiazepines. One is the sedative action, and that's what we'll look at first. If you take a wild-type mouse and you put it inside an arena, you can then track the position of that mouse as it runs around. And you can see where it goes, and you can quantify the distance that it moves. If you then give that same mouse some diazepam at a relatively high concentration, 30 milligrams per kilogram, that has a sedative action on the mouse. the mouse might just go to sleep, or it'll just relax and do nothing, perhaps staying in this corner of the cage. If you now take a different mouse, where you've made the genetic mutation so that the alpha-1 subunit is no longer sensitive to diazepam, then the basal condition, where there's no benzodiazepines present, the animal is just like wild type, the GABA alpha-1 subunit behaves just like the normal alpha-1 subunit in the absence of diazepam, and so the mouse runs around, explores its environment. You give it the large dose of diazepam, and in the point mutant mouse, for the alpha-1 subunit, it has no effect. The sedative effect of diazepam is gone, and so apparently sedation is mediated by the alpha-1 subunit of the GABA-A receptor. You can test the specificity for it acting on the alpha-1 subunit by doing the same mutation in the alpha-2 subunit. So now we take a point mutant mouse. It has the histidine replaced by an arginine in position 101 for the alpha-2 GABA-A receptor. We give the diazepam, and the sedative action still works. So sedation doesn't work via the alpha-2 subunit, but it's specifically mediated by the alpha-1 subunit of the GABA-A receptor, a remarkable specificity. The other interesting action of the benzodiazepines is anxiolysis, a reduction in anxiety. And this we can also monitor in mice by thinking about their overall behavior and what they're afraid of. And mice are worried about large open light spaces. They're nocturnal animals, and they prefer to move around in the dark. And so experimentally in the lab, we can design behavioral arenas that have dark area and a light area. And if you put the mouse in this arena, it'll tend to run around in the dark area. And every now and then it might poke into the light area, but it's afraid, it's anxious, and it'll rapidly return to the dark area. If you give these mice a small dose of diazepam, then they'll relax and their fears will be reduced. They're less anxious. And they'll then go and explore the light area to a much greater extent than under the control conditions. And that's the anxiolytic effect of diazepam, here occurring at much lower doses than the high concentrations for sedation. We can then do the same 2 point mutations as before and study the effects of diazepam. In the alpha-1 point mutation, the diazepam still works. It has the anxiolytic effect, and the mouse will explore the light area. But in the alpha-2 point mutation, the diazepam fails to have the anxiolytic effect. The point mutation here means that the diazepam can't act upon the alpha-2 subunit, and the fact that there's then no anxiolytic relief from diazepam means that anxiolysis is being mediated by the alpha-2 receptor, at least in mice. So we've seen that benzodiazepines act in a highly specific way upon some subtypes of GABA-A receptors. It turns out that the sedative action of benzodiazepines is mediated by the alpha-1 receptor, and the anxiolytic effect of benzodiazepines is activated via the alpha-2 receptor. That, of course, is an analysis that's been done in mice, and it's unclear to what extent that'll carry over into the human situation. But it has, of course, inspired the pharmaceutical industry to go and make agents that are specifically acting at alpha-1 and alpha-2 subunits, and of course it's probably also interesting to explore the other subunits of the GABA receptors that may have different therapeutic actions.